All right. So I'll just ask you to forgive me. I'm a little bit under the weather, but I'll do my best. So I was thinking about the questions that came to us when we were asked to present here, and I was thinking about replication, reproducibility, and what we're really trying to do. And in the beginning of the week, we heard a lot about the sort of hopelessness of defining behavior and associating behavior with root causes. And to me, the question is really, can we find consilience? Can we find the common outcomes of experiments associating genes and genetic loci to behaviors? And can we use that information to organize behavior, to classify behavior, and define the relationships? And so I was coming at this from a perspective of this crisis in reproducibility, which seems to occur every 10 to 15 years. As many people have highlighted earlier, the preclinical findings are not always reproduced. Many of these are leading to failures of research translation, and we did the first experiment, so we get blamed for it. But we've also seen that variation is intrinsic to experimentation and behavior, completely unavoidable. And so bioinformatics approaches give us two key solutions to these problems. One, of course, is documenting the assays and the experimental conditions that allow experiments to be replicated, reproduced, repeated. And also, they give us an approach by which we can aggregate findings from related studies to find the convergent evidence about the role of a gene or a variant across heterogeneous experiments. And these can be highly heterogeneous experiments, multiple species or multiple platforms for identifying gene-to-behavior associations. So we've heard before that there's different data from different labs, that the laboratory environment has an influence on all studies of behavior, and that these do lead to discrepancies among the outcomes that can be resolved eventually if we repeat enough tests in enough labs. Jeff Mogul and I, unfortunately Jeff couldn't be here, went through data mining of every single observation of the same behavior in his lab. It was kind of compulsive, but he did enter all these into one form, so we were able to do this. And we were able to extract what documented environmental factors in the laboratory environment had any measurable influence on this behavior. And we found that the person doing the experiment and the environmental factors had much more influence than the genotype on the trait. And that, interestingly, and Will Valder also showed this, all of these environmental factors, with the possible exception of the order in a cage with which a mouse was tested, all of them interact with genetics. And so it's a real challenge to deal with this. The context specificity of an experiment can influence its validity. So the particular nature of the environment might affect the face validity or the construct validity or even the associative and dissociative validity of a trait. We're basically saying if I measure my trait in a noisy environment, it's no longer the same trait as I've measured in a quiet environment. I've literally changed the trait. With stress-induced analgesia, the neurochemistry of that trait changes depending on the magnitude of the stressfulness of the manipulation. So all of these environmental influences might actually be altering the definition of what we're measuring. This is a replication study of genetic selection that Jonathan Flint's lab did a number of years ago. And I thought what was really interesting about this study is, you see, he did a selective breeding experiment in mouse strains, cross of mouse strains. And here he had an open field. In the first instance of the replication, it was a circular open field that was well lit. And he selected different loci than he did in 2001, where two dark open field selection studies were done simultaneously. So you could ask what changed. Was it the genetics? Was it the apparatus? But I think it was really interesting that there's both stability and instability across these studies. So how do we deal with this? We heard a lot of strategies this week. One, standardization. I think most people have dismissed this. I think John early on dismissed this because people don't want to essentially standardize. Everyone's got their way of doing the test that they feel strongly is the right way to do the test. The other strategy, of course, is to vary systematically or heterogenize the testing, and I think this is really compelling. But what if we shake things up a lot? What if we globally extrapolate this heterogenization approach, integrating outcomes of highly diverse experiments under many different conditions and test paradigms? And so there's multiple strategies that we can use to do this. We heard from Rob about a strategy we call systems genetics, doing genetic correlations of many behaviors across a panel of strains or panels of mice. And we can ask the question of which traits are correlated with one another and which ways of measuring those traits are correlated with one another, and how repeatable are those ways of measuring traits within the same population. And the integrative functional genomic strategy is a broad extrapolation of that, where we're looking at the shared association of genomic results to behaviors. And so what we're doing is taking every single set of behaviors associated with sets of genes and asking how those gene sets overlap. And we can do this in very, very large numbers of gene sets. And we don't need to use the semantics with which we label the behaviors to do this. We can actually match all of these sets of results from these experiments to align them. And so the idea is that our induction is of a higher quality if we can actually explain it through many different experiments rather than trying to repeat the same experiment over and over again accurately. We want to actually test the same hypothesis and get the same answer many times and many ways. And that's the result that's likely to hold. So there's many different resources that can be used for these types of approaches. Semantic data integration, I guess one of the most heroic attempts would be the neuroinformatics framework. We saw genenetwork.org, which looks at genetics as the basis for integrating trait data. The mouse phenome database at the Jackson Lab stores primary mouse phenotypes, protocols, data, and allows for trait correlations and an examination of the stability of trait data within and across strains. And we heard and saw several examples of people using the data from that resource. And the system that I've been developing since about 2007 now is geneweaver.org, which allows people to collect and aggregate and analyze sets of data using combinatorial algorithms to compare genomic results across species. The idea of data sharing and data dissemination, of course, facilitate the discovery of convergent evidence. But when we use semantics, we have to retrieve behavioral trait data based on the words we use to describe them. And so we get into this very challenging problem of how we describe behavior in model organisms and whether or not it can be appropriately done. So we use controlled vocabularies, a consistent vocabulary for describing information, or we may use an ontology, a hierarchical representation of concepts. But we have to ask the question, how are behaviors hierarchically related to one another? How do we organize that? Should we label them by what we think the assay means or by what we can observe? And when we talk about what we can observe, do we want to talk about a very objective type of observation that's totally free of the context and the behavioral meaning? For example, one example is immobility. Mice are immobile in a variety of apparati for a variety of reasons because we've done a variety of things to them. But the thing we quantify is immobility. Do we want to label all of those traits, depression, anxiety, fear, learning, with immobility, or do we want to describe the psychological concept that's being assessed by each of those trait measurements? So there's a lot of competing priorities in naming behavior, which is the thing that's so critical to us being able to retrieve data that purports to be measuring the same behavior. We want to avoid anthropomorphism by assigning depression or autism to a mouse. But at the same time, we want to be able to align data across species so that we can actually say, I've got a data set relevant to depression here, even though the study was done in a mouse or a rat or a yeast cell. How do we retain objectivity and also behavioral meaning with the ability to integrate assays, concepts, and context and interpretation? And so on the right, there's just an example of one of the widely used ontologies. this is just a fraction of it, mammalian phenotype ontology, that shows how behavior is currently represented. And I actually think it's a little bit disappointing when we think about the exquisiteness with which we do our experiments and define, and this isn't fully expanded, right? But we do these exquisite experiments, very in-depth concepts that we're testing, and the depth of our description of that is very poor in comparison. So we don't want to have to lean on this kind of paucity of semantics. So the other thing that we want to document, not just what was studied, but the experimental context in which it was studied. So I think Hanno had a great set of recommendations for documentation, behavioral experiments, which we tried to rigorously follow as a BXD experiment. Could we actually document all of that stuff? We call the paper the phone book. But it's got a lot of data and apparatus documentation and such. Fully structured phenotyping protocols like the IMPC use, really nice. They capture a lot of data about how each observation is acquired. And in the mouse phenome database, we have these unstructured but well-documented phenotyping protocols. So when we look in the mouse phenome database, we can retrieve, in this set of data, Walston 1 was discussed a lot yesterday, we can actually see the dimensions of the apparatus and understand exactly how each measurement was obtained. So when we find a result in a resource like Gene Network, we can go back and say, okay, these two measures don't correlate. How are they obtained? So originally, the idea of the mouse phenome database was to take a population of strains, kind of like the BXD, but a bit more diverse with a bit more idiosyncratic relationships among the strains. And the idea going into it was, we'll collect a standard set of data on standard inbred strains, and so we'll know the exact value of locomotor activity in these strains. And of course, Doug Walston recognized quite early that in behavioral studies, there wasn't going to be a reference value for a strain, that these are traits that are subject to the influence of the environment. So now we really think about this more as a platform for correlation, for studying trait diversity, trait variation, and other things. Rob just talked very nicely about the work in Gene Network, where we can integrate data across a genetic reference population. And again, just another representation of that idea. We have genotypic data, gene expression data, classical phenotypes, molecular data, and we're essentially using specialized correlations and interrogating this correlation matrix to relate broadly biological characteristics of this population across levels of scale. And it allows us to ask questions about which traits, which behaviors might be related to one another, which measurements of those behaviors, which environmental conditions give us the same values regardless of the meaning of those traits, regardless of the names of those traits, and to find the biology underlying them. And so we're really asking the question, over all the polymorphisms in mouse strains, which of them have a reliable association to a trait? We could do a similar thing in the data across the panel of strains in the mouse phenome database. Start correlating trait data, retrieved either by semantics or by the correlation to other measures. And we can ask, in the multidimensional space of behavioral traits, where does a new trait sit? Is it related, or is my version of measuring that trait related to previous versions? Is the elevated plus maze related to any other thing that we call anxiety? Yes or no? And in advanced mouse populations like the collaborative cross, the diversity outbred that Fuad's going to talk about, we have many more of these polymorphisms. And we have essentially, I guess, over 45 million polymorphisms across the entire population of mousedom. And what we really want to do is understand which of those loci, if any or all of them, relate to which phenotypes, and therefore which phenotypes share a biology, and therefore which phenotypes actually are reflective of the same construct. And we can actually identify essential biological processes and pathways associated with behaviors by running these correlations, extracting these correlations. And so we're out of the problem of classifying behavior by external manifestation. We can classify it by association. Say these are traits that are correlated, so they must have some biological integrity, and therefore the condition that we're trying to identify has a well, a biology as opposed to simply a behavioral definition that may be highly overlapping and highly difficult to discern from another disorder. So when we talk about data integration, we can lump or split, or the problem with splitting up data into lots of parts is that we end up with very little of it to analyze. We can take apart proteins, genes, mouse strains, data collected in different ways and using different technologies, and we end up with very little ability to start relating behaviors to one another because the data are so sparse. And so using this sort of monolithic block of data, lots of genes connected to lots of behaviors, we actually lose some of that data sparseness. We also lose some parametric information, a lot of parametric information, and some quantitative rigor, but we gain the ability to extract out of the diversity of data consistently observed relationships between genes and phenomena. So the idea is, in this functional genomic data aggregation, we can evaluate the relationships among many biological entities, many behaviors and many conditions, many backgrounds, many species, many environments, many experimental paradigms. All we have to do, is harmonize the biological identifiers associated with each of those molecular traits. In order to really deploy this, we wanted a nice user-oriented environment so that any biologist can access and interpret data in the system. And we're talking about a problem matching of sets to sets that in a database is an NP-complete problem. So we needed algorithms that allowed people to operate on their selected data in real time. So we, working with the computer scientist Mike Langston at University of Tennessee, were able to develop some fast algorithms for enumerating all these set intersections. And so we allow people to ask which genes are associated with user-defined concepts and constructs. The user selects a bunch of data that they're interested in, or they start with a gene of interest or a set of genes that they've derived through an experiment in their own lab, and they can extract everything that is overlapping highly with that data set. So I'm not going to go too much into the website itself, but this is basically what it looks like. We have sets of genes. They come from many different experimental types, some gene expression, GWAS, positional candidates, genetic correlations to behavior, from molecule to behavior. We also use curated annotations, so gene to disease or drug interaction to gene. So many different phenotype ontology annotations to mutants, many different ways that we can construct lists of genes with relevance to behavior or any other biological process or characteristic. And we've got now over 80,000 of these gene sets and 9 species. And through gene homology, we can align them one to another. And this allows a lot of different types of questions to be addressed. So this is a little more of nuts and bolts. Basically, we have user-deposited gene sets. You can register for an account. You can store thousands of your own data sets. The interesting social psychology here is most people store their data in their own account. And we do make it very easy to share all of that data immediately publicly so that anybody else's analysis has the ability to discover your data if you let them. You're exposing whatever data you want. And most people prefer not to expose it, so it's all sitting in this tier 5 private data. But we do have other tiers representing the level of curation of data. We align it through gene identifiers, and then we construct what's called a bipartite graph. It's basically a network-based way of representing lists. We have the name of the gene list or the function on one side, and the list of genes represented by the connections of the function to the genes. And then we can apply lots of algorithms and different tools to identify the relationships among these sets of genes. So this looks better on the screen, so that's good. So these are just a few examples of analyses that we've been able to do using this system. So take alcohol-related data sets. These are now lists of genes associated with alcoholism. And at the bottom, you could think there's individual lists We're just looking at this fraction of a large network. And at higher levels, we're looking at 2-way intersections, 3-way, 4-way, and on up. So we have 7-way intersections of alcohol-related genes. And what we've done in purple is highlight sets of genes that, or intersections of sets of genes that contain a gene that's currently known to be related to alcoholism, either through OMIM, online Mendelian inheritance in man, so human genetic studies, or mammalian phenotype, a mouse mutant exhibited an alcohol-related phenotype. And what we see, which is really interesting to me, is that alcohol-annotated genes tend to exist in each of these different experimental result sets. But if we start intersecting all of these gene sets, we find that genes that are broadly relevant to many of these alcohol-related gene sets are not currently known to have anything to do with alcoholism. And we can look at this in a number of different ways. We can also say, how do these gene sets partition? This is a single gene that we say has a broad role in the acute response to alcohol. There's a subset of gene sets that have additional genes connected to them, and another subset. So we have all of these different subtrees, if you will, or subgraphs, which each may reflect biological subsets of alcoholism, either different phenomena, different facets of the disorder, maybe acute response, maybe chronic response, maybe susceptibility, locomotor activity. And so now we want to go into these gene sets and interpret why they're grouped together. Maybe all of the experiments done in my lab through some idiosyncrasy cluster together. And I can see that here. They'll fall out. So the replicability, the positive match, is the main reason things come together here. And the dissociations, maybe they're meaningful. Maybe the meaning isn't interesting, like I'm doing my experiment wrong, so all my data sits off by itself. It's not contaminating this convergent evidence. So there's a lot of utility to this. Now, I'll just show one more applied example, how we can take many different genetics experiments and get down to a single polymorphism from the integration of this data. And this is some work that John's really motivated me to do. I think since we were working on Gene Network, he's always asking me, how is alcohol preference and withdrawal correlated? And so we built a tool in Gene Weaver, and I thought I'd try it out. So I took alcohol preference data sets. I merged them, took the union of those gene sets. I took alcohol withdrawal gene sets, and I compared those. And so one of the things I observed, though, is that there were a lot of overlapping genetic loci for both alcohol preference and withdrawal. And so we just chose one of them kind of arbitrarily and fortuitously on chromosome 8, where an alcohol withdrawal locus mapped in one population of mice, overlapped an alcohol preference locus, mapped in a totally different population. And what we can see is that the intersection of these two loci are 281 genes, a sort of hopeless number of genes to have to pick through. But we pressed on anyway. So what I was able to do then is I have the genes, several hundred in my preference QTL, several hundred in my withdrawal QTL, but we have many differential expression gene sets in the context of alcoholism. And we don't have a lot of alcohol withdrawal related gene sets, but we do have our genes that are annotated to seizure, and seizure was the endpoint in the alcohol withdrawal experiments. So I can ask, is there any convergent evidence for any genes among all of these different characteristics? And I see that there's one gene, adaptor protein 3M2, AP3M2, that is at the intersection of both QTL, and that it appears on multiple expression alcohol preference related data sets, and if mutated, creates a seizure susceptibility response to pharmacological agents. So that's exciting. So luckily, I was able to find mice, because there's a nice database of mice. And there was a mouse in Hiroshi Ono's lab in the Riken Institute in Japan. I was able to get that imported at Jackson Lab. And we were able to show that this alcohol mutant has an abnormal withdrawal response to alcohol, and it also has a heightened preference for alcohol at an interesting dose, about 9 mg per kg. We see this pharmacologically relevant preference to alcohol. Problem is there's no polymorphisms in the gene, so we were a little bit upset by that. But again, we've got all of this genetic data across many different populations, so we may actually be able to find it anyway. So there's no genetic polymorphism in the gene. So the first thing I did is went to Gene Network and said, well, maybe there's an expression regulatory polymorphism. And of course there is, because I was incredibly lucky when I was doing this. So there's this nice narrow expression QTL for AP3M2. I've got the alcohol preference locus from Bachmanoff and Tordoff's lab. And then I've got the original alcohol withdrawal locus that was mapped by Susan Bergeson and John Belknap and others. And so now remember, we're honed in on this gene already because we had all this functional genomics data from the gene expression data sets. And we, without going into too much genetics, had to reconstruct the backgrounds of the 8 strains that went into the heterogeneous stock that was crossed to map this withdrawal locus. So we actually were able to say now, what SNPs are segregating, what polymorphisms, allelic variants are segregating at the location of the intersection of these QTLs. And so we were able to infer now what polymorphisms could account for all of these traits having the same regulatory locus. They don't necessarily have to, but we wanted to start from that assumption. And so we were able to refine this by looking at mouse phylogeny. We have withdrawal seizure prone mice. We have the black 6 mice, which are also having a high preference phenotype. And then we have low preference, withdrawal seizure resistance, and low abundance of the AP3M2 transcript. And we were able to narrow that down. Again, we're looking for an expression regulatory polymorphism, so we went to a database of gene regulatory regions. We found only one that's in neural cells. And then I said, OK, so now we're down to 11 SNPs. And I went to the cafeteria and explained my troubles to Ken Pagan, a noted geneticist. And he said, well, you know, I have this DNA methylation data upstairs. And I thought that was good. We were able to show that only one of the 11 SNPs in the neural site is actually an epigenetic modifier of the expression of AP3M2. So by aggregating lots of evidence sources, we were able to show that we have a polymorphism that's influencing both of these related traits, just starting from a search of genomic data associated with these traits. So, you know, I was thinking about this. We do this massively parallel genetic experimentation, and you know, Karl Popper urges us to do these falsifiable experiments that are excluding other alternative hypotheses, that we really can't have a system in which we can derive any possible conclusion out of our experimental analyses. So when I heard Wim, I thought, it's a hopeless situation. We can say anything we want to out of this data. But what I really think about is, you know, we want to evaluate the associations among diverse experiments en masse and enable the detection of the conditions under which they associate and the conditions under which they dissociate. So if we think about a new, and I don't know if it's a new paradigm or if or not, but really the inference is whether or not these data have cohesiveness. If we classify the experiments by their results, we can then ask what the experiment was, rather than assume that we're reducing all these other explanations. So we're just sorting out the experiments. And the drivers of that similarity and dissimilarity could be environment, could be paradigm, it could be the intrinsic variation in behavior itself. So in summary, variation across behavioral studies, it's unmitigated by our best efforts to do anything reasonable. Although I think Yoav's got some really interesting ideas about estimating gene by lab environment and doing a more refined job at finding replicable results. These data integration strategies that we're using help us find convergence across assays. and measures of the same behavioral construct. So we can actually find the shared associations to allelic variants. Our genomic experiments can be classified by results to define the commonality of what was tested. So we don't have to know what it means. We can parse that out of what the results were, and go figure it out later. Yes, it's funny. And so there's lots of challenges, right? We don't really describe our experiments very well. We don't document and share data very well. And of course, we need more algorithms, tools, and approaches for trying to sort out these types of experiments. It takes a village. Some of that village is here. A lot of the genetics work in my lab was done with Vivek Philip. Jeremy Jay was one of the lead developers of GeneWeaver over the first 7 or so years of its existence. Jason Bouvier has done a lot of the alcohol experiments with the AP3M2. Some others in the lab involved in some of the other studies that we're doing. Molly Bogue actually runs the Phenome Database project in my group. Steve Grubb's the developer on that. And Eric Baker and Mike Langston. Eric's at Baylor University, Mike at University of Tennessee, Knoxville. Both of them are excellent computer scientists who've been working with me carefully on the development of GeneWeaver, which we've been enjoying together for many years. And so some funding sources, NIAAA and NIDA supporting this work. So thank you.